What is the role of jumping genes in genomes? What triggers their jumping, and why are they almost never found in asexual organisms? We will do our best to try to answer these questions, as well as other similar and related questions, in today's episode of the Secrets of Genes series, to which I warmly welcome you. My name is Edward Kejnovsky, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. What is the role of transposons? At first, everything seemed clear. They are simply parasites and can only harm their host. The explanation sounded quite logical, as their jumps represented a powerful mutagen. Indeed, a number of diseases caused by the insertion of transposons into or near an important gene have been described. Now, however, it turns out that the situation is not as simple as scientists previously thought. The idea of transposons as primary parasites is not incompatible with their significant role in genome evolution, and evidence of their usefulness is rapidly increasing. It is becoming increasingly clear that they are key players in evolution. There are known examples of so-called transposon domestication, where a transposon is used for a function beneficial to the host. The ubiquity of transposons likely reflects some general process, an important role for these companions of ours. Not only does it prove their antiquity, but it likely testifies to an inherent need for the genome to contain mobile segments, allowing organisms to respond flexibly to changing external and internal environments. Asking about the usefulness or uselessness of transposons is the same as asking about the usefulness of any other genetic mutations. There are good ones and bad ones. Without them, there simply would be no evolution. Transposons are activated by external stress, which is especially evident in plants, which, unlike animals, cannot escape a stressful environment. Temperature, drought, radiation, or pathogen attack lead to transposon activation, and their multiplication then causes destabilization of the entire genome. Genome destabilization and rearrangements potentially increase an organism's chance to adapt to changed conditions. Through the amplification of transposons, the genome essentially creates the conditions for its own rearrangements and improved adaptation. Genome rearrangements are, of course, passed on to future generations. Genomes are inherently unstable systems. They are generators of their own rearrangements and other changes. In which cells and at which stages of an organism's life does transposon jumping occur? From an evolutionary perspective, only new transposon jumps in the germline are significant. Parts of the germline are actually somatic cells that eventually give rise to germ cells, and thus they pass into future generations like endless rivers of genes, flowing steadily through the generations. Just to give you an idea, in the most numerous family of retrotransposons in our genome, the so-called ALU elements, of which there are 1.5 million copies, a new insertion occurs in every 20th newborn individual, and in ancient human ancestors, a new insertion occurred in roughly every single individual born. Each offspring therefore had, on average, one more ALU element in their genome compared to their parent. Recently, however, it has been shown that transposons jump around our genome not only in the germline, but also in somatic cells, that is, in the cells of our bodies that are not passed on to offspring, in cells that die with us. A sensational finding was the recent revelation that during human brain development, thousands of jumps of the L1 and ALU retrotransposons occur in neurons into new locations in the genome. Scientists use state-of-the-art sequencing technologies and map the sites where the transposons were inserted. They even managed to read the genomes of individual neurons and compare the retrotransposon jump sites within them. The study showed that our brain is a kind of mosaic made up of genetically distinct neurons. Are these differences in neurons related to their different functions, to their specialization? Transposon activity apparently accompanies the normal development of neurons. The question is whether new transposon insertions into certain genes could also be related to developmental abnormalities and thus lead to neurological or psychiatric diseases. Other research teams have shown that transposon activity is also significantly increased in cancer cells. Asexual animals provided a surprise for scientists, specifically bdelloid rotifers of the order Bdelloidea, a group of species that have lived without sex for tens of millions of years. It turned out that they contain very few transposons. They don't contain aggressive retrotransposons, but only less harmful DNA transposons. Even in plants, it seems that more sexual species contain more transposons. For example, the genomes of cross-pollinating plants carry more of them than the genomes of self-pollinating plants. Why is that? The answer likely relates to the fact that sexuality allows for the repair of naturally occurring DNA errors through recombination. The non-recombining genomes of asexuals are, for these very reasons, prone to the accumulation of transposons. Without recombination processes, the genomes of asexuals would soon be completely infested with transposons. Once these species abandoned sex, they had to get rid of retrotransposons very quickly. They had no choice. Those species that didn't do it simply went extinct. Some scientists have even made very bold claims that transposons actually caused the emergence of sexuality so they could spread more easily between species. The most significant function of transposons from a genomic perspective is their mutagenicity. They are powerful endogenous mutators. Mutagenicity is an inherent property of theirs. They generate mutations of all kinds and of various scales. Transposons can jump into a gene and thereby disrupt its function. Because there's a significant number of transposons in the genome, mutual interactions and exchanges of genetic material can occur, leading to so-called DNA recombination. The results are then deletions, inversions, or duplications of large regions of the genome. DNA rearrangements generated by transposons are apparently random or stochastic, yet regulated. You might argue that chaotic or destructive processes caused by transposons must always be harmful or even fatal to the cell and the organism. However, we must view everything from the perspective of long-term evolutionary processes, where the described processes represent key mechanisms for the gradual building of complex systems and structures. Let us not forget that even very rare but currently advantageous genome arrangements can indeed pass through the sieve of natural selection. That would be all for today. In the next episode, we'll talk about how jumping genes can be useful for the host organism. I'll be looking forward to seeing you. If you like my storytelling, I'll be grateful for a like, and if you want me to continue with episodes on genetics and evolution, please subscribe, which will help these videos reach others. I really appreciate your support. Thank you, and bye-bye.